Yes, welcome to today's session. So today I would like to discuss with you some of the important points and important diagrams from the topic called Plasmodium vivox and its life cycle in the various aspects. Now let us see Plasmodium vivox. Basically, let us learn some introductory points. Introduction is not required to write in the examination. Plasmodium vivox life cycle is important for 8 marks question, especially in the examination point of view. So it was divided into basically sexual phase and asexual phase. In the examination point of view, either sexual phase will be given or asexual phase will be given. Both are important for the examination point of view. So that's why any one, either asexual or sexual phase will be given in the examination part. So the Plasmodium vivox, basically it is a digenetic parasite. It completes its life cycle in 2 hosts. Okay, that's why it is called digenetic parasite. Okay, so basically it completes its sexual phase, sexual reproduction in female Anopheles mosquito, whereas it completes its asexual life cycle in a man. So these two are the hosts for the Plasmodium vivox and its life cycle. Okay. Now let us continue asexual life cycle of Plasmodium with the help of neat label diagram and points itself. If you take the asexual phase of life cycle in Plasmodium, it is commonly called schizogony. What is that called? Schizogony. Now, the schizogony was classified into basically 2 different types. The hepatic schizogony, the first one, and the second one is erythrocytic schizogony. If you take the hepatic schizogony was divided into 2 major types. Pre-erythrocytic schizogony, you can see in the flow chart, and the flow chart you have to mention in the diagram in the examination part also, and also exo-erythrocytic schizogony, that is the next one. Next, if you see the life cycle diagram, it looks like this. The asexual phase of Plasmodium vivox in human or man. So diagram is very important, and you can take the diagram in the given part. You can see the schizont, how it looks like. Okay, here you can see cryptozoites. You can see the cryptozoites here. Okay, imagine that when the infected mosquito, female Anopheles mosquito, sucks the blood from a healthy human. Is that a healthy human? What happens? It releases some of the sporozoites into the blood. The sporozoites, whatever enters into the human body, they reach to the liver cells for the further developments. So the hepatic schizogony, nothing but sporozoites undergoes for further modifications inside the liver cells and complete their life cycle, one part of life cycle in the liver cells. Hence, it is called hepatic schizogony. What is that called? Hepatic schizogony. Here, tropozoites are produced from the sporozoites. When the sporozoites reach to the liver cells, they transforms into a tropozoite. The tropozoite contain a blunt finger like a pseudopodia, and the tropozoite stage generally called as this stage is called as a schizont stage. What is that called? Schizont stage. The nucleus of schizont undergoes for multiple divisions and able to produce special cells called cryptozoites or first generation merozoites. You can see here cryptozoites. Okay. The cryptozoites are also termed as a first generation merozoites. Why? Because they are belongs to first generation. These first generation merozoites, some of them here attacks the fresh liver cells and continue another hepatic schizogony. But some of the first generation merozoites or cryptozoites continue exo-erythrocytic cycle. Now what happens during exo-erythrocytic cycle? Let us see with the help of points. I told you already, hepatic schizogony is the first stage. Point by point let us learn. This was discovered by Short and Gurnham. Point number 2. Whenever the infected mosquito bites and sucks the blood of man or human, it releases 2000 sporozoites into the blood of human through its saliva. The mosquito saliva, through that these sporozoites enters into the man. You know very well about the sporozoites. In the previous video we have seen a structure of sporozoid, and these 2000 sporozoites within half an hour, what they will do, they reach to the hepatocytes, nothing but the liver cells. There undergoes for further modifications. So what are the further modifications and the developmental changes occurs in this structure? We are going to learn in pre-erythrocytic schizogony and the exo-erythrocytic cycles. Now let us start with the pre-erythrocytic cycle. Whenever the sporozoites reach to the liver cells, they transforms into tropozoite stage. The tropozoite stage feeds upon the content of liver cells. The liver cells contain various types of nutrients like proteins and glucose, etc. Highly active nutrients are present in the hepatocytes. These content or nutrients, whatever present in the liver, utilized by this tropozoite stage and assumes spherical shape, attains maximum size in this stage by feeding upon the content, and this stage is called as a schizon stage. What is that called? Schizon stage. Its nucleus divides several times. The nucleus, whatever it has, divides several times. Mitotically, the nucleus undergoes for several divisions. Along with the nucleus, cytoplasmic divisions are also takes place, resulting in the formation of approximately 12,000 daughter cells. What are they? 12,000 daughter cells called cryptozoites or first generation merozoites. These cryptozoites are first generation merozoites. By rupturing the sinusoids of the liver cell membrane, they reach to the liver cells directly. In between the liver cells there are sinusoids. So the sinusoids are on the outermost layer covered by cell membrane. So this cell membrane should be ruptured, and finally after rupturing, they make a passage to enter into the liver cells. That is the meaning of that rupturing. Now this entire process is completed approximately in 8 days. 8 days are required to complete this process. Now this first generation merozoites, they have two options. Either they can enter into the fresh liver cells to continue exo-erythrocytic schizogony, whereas they can enter into the RBC to continue erythrocytic schizogony. If they enter into the RBC, they undergoes for erythrocytic schizogony. Now exo-erythrocytic schizogony, how it happens, let us see. If the cryptozoites, whatever there, let us take the cryptozoites, if they enter into the fresh liver cells, they undergoes similar to the changes that are occurring in the pre-erythrocytic schizogony. Okay. Finally they produces, now here second generation merozoites will be produced. Okay. These second generation merozoites are classified into two types based upon their size. Some of them are larger in size, some of them are smaller in size. These second generation merozoites are called metacryptozoids. What are they called? Metacryptozoids. Classified into micrometacryptozoids and macrometacryptozoids. Now, this entire process is completed approximately in 2 days of time. How many days it takes? 2 days. Macrometacryptozoids, if you take larger in size, they enters into the fresh liver cells again and continue another exo-erythrocytic schizogony. Whereas micrometacryptozoids are highly infective to RBC and they attacks the RBC cells and continues erythrocytic schizogony. Now how erythrocytic schizogony happens, let us see. Erythrocytic schizogony was for the first time explained by Golgi. You know Golgi? And hence this is also known as Golgi cycle. What is that called? Golgi cycle. Now the cycle is initiated by entry of microcryptozoids, metacryptozoids from the exo-erythrocytic schizogony and cryptozoids, entry of cryptozoids which are produced in pre-erythrocytic schizogony also causes initiation of erythrocytic schizogony. In the fresh RBC cells or red blood cells, the structure, these structures assumes spherical in shape and transforms into tropozoid Now, it develops a small vacuole. Imagine this is a small vacuole which gradually enlarges in size day by day or hour by hour and pushing the cytoplasm and nucleus to the peripheral region. When the nucleus has gradually increases, it pushes the nucleus towards the peripheral region. Now the plasmodium looks like a finger ring. Plasmodium looks like a finger ring. Hence this stage is called as a generally signet ring stage. What is that called? Signet ring stage. Now soon it loses the vacuole, attain pseudopodia. Vacuole will be lost, attains pseudopodia. It attains pseudopodia and loses vacuole. Now this stage is generally called as a amoeboid stage. What is that called? Amoeboid stage. Why? Because it looks like amoeba. It develops a pseudopodia. With the help of pseudopodia, it actively feeds upon the content of RBC. Whatever the nutrients present in the RBC utilized by this amoeboid stage of Plasmodium and increases in size or enlarges in size, assumes the similar shape to the amoeba, and RBC now grows double in size. RBC grows now in double in size, and this process is called as a hypertrophy. What is that called? Hypertrophy. Now the material here, whatever you see inside the RBC, that contain various types of nutrients. Now you know RBC also contain hemoglobin, right? What is that? Color giving substance called hemoglobin. Now malarial parasite digests the globulin part of hemoglobin. What is globulin part? You know very well about it. The globulin part will be digested by the protein part of the hemoglobin digested by the malarial parasite and converted into insoluble substance that is called as a hemojoin. What is that called? Hemojoin. Due to hemojoin, you may get a chilled fever. What is that? Chilled fever. After this, it is the malarial pigment, which is a disposable product. The hemojoin is called malarial pigment. For MSET also continuously they are asking the bit, what is a malarial pigment? It's definitely hemojoin, which is a disposable product. During this stage, small red colored dots appears in the cytoplasm of the structure. This cytoplasm which contain red colored dots are called Schaffner's dots. What are they called? Schaffner's dots. These are believed to be the antigens of RBC. Antigens of RBC. Now, RBC becomes a schizont. RBC become a schizont, undergoes to schizogony. Undergoes to schizogony. Similar to that of pre-erythrocytic schizogony. And produces approximately 12 to 24 merozoites. Now the cell membrane ruptures, bursts and releases these 12 to 24 merozoites into the RBC, into the blood. So like this, it completes its erythrocytic schizogony. Through these merozoites later on, microgametocytes and macrogametocytes will be produced. From the microgametocytes, generally, if you take the microgametocyte, male gamete will be produced. From the macrogametocyte, female gamete is able to develop. This is the end for asexual mode of reproduction in Plasmodium. Remaining part we will see in the next class. Thank you.